This is Data Observatory, which is one of the biggest visualization platform in UK. It's also one of our main analytical tools for multidisciplinary study in the DSI. So we built this one about 1 year ago, which now is heavily used in the various multidisciplinary research, including the medicine work, neuroscience, and earth science, astrophysics, many areas. So one of the particular focus in DSI is to apply data science to various disciplines. Medical and healthcare is one of our key focus. And things about back to the 5 years ago, we already started a big project with Roy Brompton Hospital and the National Heart and Lung Institute on the project for the respiratory disease. The project called U-by-Pred, and it has been a very big success for applying data science to the medical research. So today I'm very happy to have here with our distinguished professor in this area to talk about how they are experienced to use this platform and working with DSI to carry out their research work.
U-by-Pred is a big European project that was funded by the Innovative Medicines Initiative for the application of systems medicine to severe asthma. And this funding was acquired because severe asthma is a major clinical problem, because these represents patients with asthma that do not respond to the treatments that we now have available for asthma. And also because severe asthma is a disease that's very heterogeneous in its presentation. Therefore, the systems approach medicine to severe asthma would allow us to phenotype the disease and also find new treatments for severe asthma. And in order to achieve those objectives, we've been very lucky to work with the Data Science Institute here at Imperial College, where all the data that was gathered during the U-by-Pred research project, a lot of bioinformatic analysis was performed.
Transmat is a powerful platform for biomedical data management. It is a tool which basically allows us to efficiently store and manage huge amounts of information, what we refer to as big data. It is powerful because it allows us to work collaboratively, and it enables our efforts towards precision medicine. So basically, Transmat allows us to select subgroups within our population and identify patients that we are interested in, which is very important. For example, we could choose age, and then we could select the population that is below the age of, say, 56, and we see our distribution in terms of gender, ethnicity, and also our clinical cohorts. Using the main clinical features which were chosen by our key leading scientists, U-by-Pred has produced a clustering of the population based on clinical features, which is exactly what we are looking at here. So we have 4 clinical clusters, and we can see the genes, the expression of the genes in each cluster. So what we are looking at here is another representation of our clusters. Since we've selected patients below the age of 56, we can see that only 34 males are now included in our cluster and 46 females in cluster 1. In addition, what we are able to do on the fly is to have a quick look at the genes which are associated with each cluster. So if I select, let's say, CCL22 from cluster 2, immediately we get a view of the Genome Browser where we can see exactly where the gene is located on which chromosome, and we can also have a quick look at the protein coding regions. In addition, we have the capability to look at molecular pathways. So Transmat is showing us here the 3 molecular pathways in which this gene is involved. Interestingly, the first one is the JAK-STAT signaling pathway.
So now I'm going to present to you some of the molecular phenotyping data that we've had from U-by-Pred. So I will present to you the molecular phenotyping that we've obtained by looking at sputum cells. And so what is shown here is in fact the three clusters that we have obtained just by looking at the genes expressed in those sputum cells. And you will see that the hierarchical clustering here gives us three clusters, which we have termed TAC1, TAC2, and TAC3. So if you look at TAC1, one of the top genes that characterizes TAC1 is IL-1 RL1, which is in fact the receptor for IL-33, which is present on many cells, but the activating cytokine IL-33 is produced mainly from the epithelium and is one of the drivers for Th2 inflammation. TAC2, on the other hand, is characterized mostly by genes that link up with inflammasome activation. For example, IL-18RAP, which is the receptor associated protein, and of course also caspase 4. While for TAC3, the genes that characterizes TAC3 are genes that are associated with mitochondrial metabolic changes and ubiquitination, and therefore TAC3 is associated with mitochondrial oxidative stress phenomena. So how do those TACs link in terms of the inflammation that we can measure in the sputum? So here we see here the distribution of the percentage of eosinophils in the sputum in the three TACs, and we see immediately that TAC1 is characterized by an eosinophilic inflammation. So it's an eosinophilic associated cluster associated with the increase in IL-1 or L1, which is the IL-33 receptor. While TAC2, which is associated with the inflammasome activation genes, we can see here that it's associated with neutrophilia. Very, very clear-cut increase in neutrophil counts in the sputum compared to TAC1 and TAC3. On the other hand, TAC3 is not associated with any increase in neutrophils, neither with an increase in eosinophils. So it's associated mostly with post-granulocytic inflammation. But in some of those patients, there is a bit of eosinophilia appearing here. So now Professor Jan Adcock is going to present the analysis using a different approach from the hierarchical clustering, which is a topological data analysis.
Okay. As you can see in the maps over here, there is some spread in the number of patients within each group. So we can use alternative approaches to analyze the data to try and define those subgroups of patients within each TAC. So here we're using topological data analysis, which looks at the structure of the data rather than the individual data that we see on the heat maps, to try and explore what might be driving those different groups of eosinophilic TAC1 patients, for example. So here I've highlighted in red the highly expressing eosinophil patients, and we can see on this structural map that there are at least three groups, one group here, one group here, and one group here, that form a different structure, and potentially this is due to a different mechanism that drives the high levels of eosinophilia here. So we've gone to subsets of the highly expressing TAC1 patients. And these can be linked directly to clinical features or to gene features in the same way that we have for TAC1. And you can see that we can look at neutrophils or macrophages in exactly the same way by highlighting the patients who are highly expressed or they have particular cell types to try and give us subphenotyping of these patients. So that's an alternative approach from the traditional approaches that we've also taken here. One of the issues we have with the complexity of data is how to make predictive models. And this is one of the challenges that UBioPred gave us, was to make a predictive model. So as we can see on this screen over here, we're starting to make an asthma map So this is trying to integrate all the data we have from the public databases and also our own data that we've obtained through UBioPred to start to integrate the numbers of genes, the proteins, the lipids and other reactive molecules that can drive the severe asthma. And one of the sub-projects of UBioPred is to make mathematical models that will predict if we remove IL-17, for example, that comes up in some of these subsets of severe asthmatic patients, what would be the overall effect of that on the asthmatic patient. So we can get potentially predictive effect, beneficial effects, but also potential side effect profiles from these maps or asthma disease map. One of the other features we're trying to look at is how does this expression of genes relate to the DNA that people have, the genetics that we know is one of the markers that highlights the asthmatic patients and distinguishes them from healthy subjects and patients with other chronic diseases. Here we have a Circos diagram looking at the expression pattern of all the genes in blood compared to the genetic profile of this. So if we look at chromosome 17, for example, which we can see is important for driving particularly childhood asthma, we can see that there are a number of genes associated with polymorphisms here. And some of these are linked to those that are associated with genetic drivers. But in these adults, we see less of a drive in blood with gene expression than we see in sputum. So in sputum, what we see is that genes such as IL-33 receptor, the R1, RL1 in TAC1, are very highly driven by the genetic component in those particular patients.
So you have seen the power that Transmat has in helping us in defining the various fingerprints of severe asthma, and also to try and find out the various biomarkers that are associated with each of those fingerprints. And obviously, down the line, we will be able to find targets that might be useful to block in order to find new treatments for this group of severe asthma. So all in all, we are gradually achieving our ambition to provide personalized medicine for patients with severe asthma.